Carbon in, carbon out. Balancing food webs with Ecopath. For much of human history, people have relied upon marine ecosystems for food and other resources. However, in recent times, pressures caused by humans such as climate change, pollution, and overexploitation of these habitats have greatly threatened our continued use. Ecosystems are defined as interconnected systems of biological organisms, which interact with each other and the environment. Ecosystem modeling is the use of models to describe these interactions, helping scientists predict the outcomes of these complex interactions in a meaningful way. So what is Ecopath? Ecopath is a form of ecosystem modeling that creates a mass-balanced snapshot of the ecosystem using different biomass pools. These biomass pools can represent a single species or a group of species with similar roles. The Ecopath master equation is as follows. The production of the ecosystem equals the catch plus the predation mortality, biomass accumulation, net migration, and other mortality. Utilizing the Ecopath model can greatly increase our understanding of an ecosystem and help influence future management decisions of industries such as fisheries in the area. A simplified Ecopath spreadsheet model of Smith's Lake was made using data collected on the BIOS3081 field trips over the past few years, as well as the DPI's fisheries data. Here is a map of Smith's Lake. It is an intermittently open and closed lake or lagoon, also known as an icol, located on the mid-north coast of New South Wales, Australia. Usually closed from the open ocean by a sandbar, it is periodically opened either naturally by erosion or by the local council with a bulldozer whenever the water level of the lake climbs too high due to rainfall. As of April 2024, the lake was last opened in September 2022 and has been closed since February of 2023. In our model, we used 15 different ecotrophic groups. These included seabirds, large fish, small fish, herbivorous fish, benthic fish, cephalopods, benthic crustaceans, zoobenthos, such as snails and such, epifauna like this amphipod over here, sessile filter feeders like sponges, jellyfish, and zooplankton, and for primary producers, we used phytoplankton, macroalgae and seagrass in one group, and detritus as the third group. The relationships between different groups are shown using this diet matrix. This matrix was based on observations and logical assumptions, such as herbivorous fish eating mostly macroalgae and seagrass, as well as analyzing the gut content of things like small fish, where we found that they mostly ate zooplankton. From this matrix, we can see that 60% of the seabird's diet is small fish, and that zooplankton is prey to the most groups. We can also see that seabirds and large fish eat the most amount of different groups. Here is the Ecopath spreadsheet model of Smith's Lake. There are 4 input parameters for the model. Production to biomass ratio, which is how quickly the organisms grow and reproduce in biomass per year. Consumption to biomass ratio, which is how much food the organism consumes in their body weight per year. Ecotrophic efficiency, which is the amount of energy kept in the system for that group. And harvest, which is the amount of biomass caught for human use per year. Utilizing these 4 parameters, we can calculate each of the parameters in the Ecopath master equation and solve for how much biomass groups like small fish or zooplankton is needed if we assume the ecosystem is balanced. This is shown in the calculated biomass column, which is incomplete. Biomass was also measured using various different methods for different ecotrophic groups. For example, we used DPI fisheries data for large fish, and for small fish, we took fish samples using the beach science method, which we then took pictures of with a ruler, and using length to weight conversions, we calculated their biomass. The measured biomass can then be compared to the calculated biomass to evaluate if the measured biomass is enough for a balanced ecosystem. For example, we found only 56% of the required small fish biomass for the ecosystem. This was probably due to flaws in our sampling methods, as we could only sample fish close to the shore and not fish in the middle of the lake. Also, we only found 30% of the required zooplankton biomass, probably due to much of the epibenthic zooplankton not being able to be measured by plankton nets or the optical plankton counter we used. Also, since we only sampled during a few days of March, the results could have been affected by weather conditions such as heavy rainfall experienced during sampling. In another example where Ecopath modeling is used, Zhang et al. Modeled artificial and natural reefs in the Yellow Sea to find if there are any differences ecologically between natural versus artificial reefs. Here is a map of the study location. The study area is in the North Yellow Sea, off the shores of the Shandong Peninsula in China. For the model, they used two different diet matrices for the artificial and natural reefs. Notice there are 18 different groups instead of the 15 we used for Smith's Lake, and some of these groups are single species. For example, Crassostrea gigas, also known as the Pacific oyster. Like our Smith's Lake model, they also had three different primary producer groups, namely phytoplankton, algae, and detritus. Some of Zhang et al.'s findings included that artificial reefs were less mature than natural reefs, with a higher total primary production to total respiration ratio. This ratio is often used to measure ecosystem maturity, with the ratio closer to 1 indicating a greater maturity. A large ratio usually indicates an immature system, with a lot of the primary producer biomass not being fully utilized. As we can see from the model, the biomass of the Pacific oyster is much greater in the natural reef than the artificial reef, at 65.19 tons per square kilometers versus 28.59. These oysters consume mostly phytoplankton, which may help to decrease the ratio. Also note that the consumption to biomass ratio is denoted as Q over B instead of C over B in this model. Another finding of the study was that both the artificial and natural reefs have similar stable trophic structures. As can be seen in this diagram, most of the biomass and energy is found at the mid to lower trophic levels, namely in phytoplankton, algae, and detritus, as well as in oysters, infauna, and zooplankton, all of which consume these primary producers. In conclusion, ecosystem modeling, such as with Ecopath, is a great way to model the complex interactions between different ecosystem groups. It's a great way to predict the flow of carbon from primary producers such as phytoplankton all the way up the trophic ladder to top predators such as sharks and dolphins. These models are a useful tool for informing the management of fisheries, such as with the helping of understanding the effectiveness of artificial reefs.